Hey folks, why don't we get to work here? Have you guys heard the big news about Wright State?
Yeah.
We're hosting a presidential debate, not just a presidential debate, the first presidential debate. So 1 year from now, you guys are going to be proud to be Wright State affiliated at the very least. I want to make sure you all get this message out, right? When somebody in Dayton asks you if it's cooler to host a presidential debate or the play-in games for the NCAA basketball tournament, the answer is presidential debate, right? University of Dayton has the play-in games for the NCAA tournament, but we've got a presidential debate, so that's something we should be pretty excited and pretty proud of. I've had people, I've told you guys, I spent last year at Notre Dame. People at Notre Dame are asking me now for tickets, right? They're asking me if I can get them hooked up with seats a year in advance for that presidential debate. So I think that's pretty cool.
Can you?
All right. So, did you ask, can I? I hope that I can. I'm not sure if I can. I haven't actually tried to I know who to ask, but I haven't tried to get tickets yet myself. I'm sure there's going to be room. It'll be in the Nutter Center, so I'm sure students will be able to get in without any pull from some little old faculty guy like me. So, hey, now for the buzzkill, right? Here is the big news for this class. 1 week from today, there's going to be a test in here, all right? Get here early because the seats are going to be hard to come by. It's going to be packed. It's a multiple choice test, just like the one in the back of your lecture notes, just like the ones that you'll find online in Pilot for the course. 45 questions, all multiple choice. Bring a number 2 pencil so you can bubble in the correct answer on a Scantron form that we'll give you. You don't need to worry about this right now, but I'll tell you now anyway. It'll save me a few seconds at the beginning of the exam. You can keep the test. All that we want from you are your Scantron forms that have the answers on them. So you can take the test home with you, mark what you think is the right answer, and then you'll be able to compare. You'll be able to grade your test before we do because you'll be able to compare your answers to the answer key pretty much right after the class is over. Right? So all we want is the answers. You take the test home with you as a memento of your exciting Thursday morning next week. 45 questions, all multiple choice. For goodness sakes, look at the old exams on Pilot. That's going to help you a lot. The more time you spend looking at those, I think the better off you're going to be. And there's something about this class. You guys are juicing me up in a special way. I've been caffeine-free for a while, so I don't think it's anything like that. But you guys got me excited and talking faster than I normally talk. All right? And what that's translating to, excuse me, is I'm a little bit ahead of schedule in terms of what I want to talk with you about in this class. And so what that translates to is this. Excuse me. What I want to try to do today is talk about not just the rest of the lecture outline that I started on Tuesday, but I also want to finish talking with you, if I can, what I was going to talk with you about on Tuesday of next week. So today I'm going to try and finish off all the lectures that are going to be covered by that first exam on Thursday of next week. And then you ask, what are we going to do with all of this time in this beautiful room on Tuesday morning of next week? Here's what I propose. You guys know it's entirely optional for you to come here anyway, right? I'm not taking attendance. There are no quizzes. It's all available through Tegrity on Pilot. So if you don't want to come on Tuesday, that's cool. I won't be hurt. Don't worry about it at all. But if you do come on Tuesday, what I'm going to do is this, is I'm going to give you a Reader's Digest version of each of the lectures up to this point in time, maybe spend 5 minutes on each, right? I'm going to flash the outline for the lecture, and then I'm trying to limit myself to just 2 images from each lecture. And I'll flash the outline, I'll flash a couple images that'll help me illustrate what I think are some of the major points from that lecture. And with the time that we have left, probably on the order of an hour on Tuesday, after I've done my little Reader's Digest spiel, I'm just going to take any questions that you have, right? Anything at all. I don't mind if you ask me the same question 6 times. I'm very patient about that sort of thing. I don't mind if you only come here for the last 10 minutes of the class. I'll be here the whole time. If you come the last 10 minutes and you ask me things that people asked me about an hour earlier, I'm not going to chastise you or say, oh, well, if you were here earlier, no, I'll just answer your question. And the people who heard me answer the question before will marvel at how similar my second rendition of the answer is to my first. Right? I'll work hard to stick to a party line. So that's the deal. I want to run through everything I was going to talk with you about on Tuesday's lecture today, and then Tuesday instead will be something of a review session. Right? Entirely optional. My ambition is I want you guys to knock this first test out of the park. I really want you guys to do well on this first test. I am pulling out all the stops that I know to pull. If there's other things that I can do, I want to know about it. But I got all those tests available for you. I've been poking you and prodding you to look at those old tests in ways that I've never poked and prodded a class like this before. We're going to do a review session. Instead of a class, the one before the exam, again, I'm doing everything that I can think of to help you guys do well on this first test. And what I'm hoping I can do is compare the class average, your class average, to previous year's class averages and see if some of these things that I'm doing are having an effect. All right? So, test a week from today. If you haven't looked at those old exams, goodness, this weekend is the time to do that, all right? And then on Tuesday, we'll talk some more, just as like a review. Thursday, we'll have the test. The following Tuesday, and that's all we're doing on Thursday, by the way. You're only taking a test. When you're done with the test, you leave. The following Tuesday, we'll start talking about thermodynamics, for lack of a better word. I'm not really excited to say that we're going to talk about thermodynamics. I'll tell you why I'm not excited about thermodynamics when we get there, but back in the swing of things a week from Tuesday. Okay. As you can see from what's been hanging behind me all this time, there are some supplemental instruction sessions that may be very popular in the next couple of sessions that meet. And so they're Tuesdays and Thursdays, 3:30 till 4:30. That's the room. Lauren would be delighted to see you there and answer any questions that you might have. Any questions for me about what's going on right now? So let's just do it. I started with you on Tuesday talking with you about the anatomy of cells. And I tried really hard to emphasize to you this idea that in biology, at IB level, structure and function are intimately related. And that ends up being the justification in my mind for taking the time to talk about these structures that comprise the anatomy of cells. Okay? Because these structures in turn tell us something about their functions, the functions of these cells and their capabilities, and also tell us something about why these structures exist in the first place. So I started with talking with you about just this general idea of the difference between prokaryotes and archaebacteria that look superficially very much like prokaryotes and eukaryotic organisms. Right? That's where we started. There's a picture for you again of a fairly stereotypical kind of prokaryotic organism. When I look at a prokaryotic organism, a picture or an electron micrograph like the one behind me, first word that comes to mind to me is simple. Now, when you're looking at it, you may think, oh, but if it's so simple, why are there all those squiggles? Why are all those names there? Bottom line is compared to what's on the next slide, a eukaryotic cell, this is simple. I wasn't kidding when I said a prokaryotic cell is pretty well described as a bag of goo. The bag is a plasma membrane, and inside, one of the main things that's hanging out in the goo that makes the goo special compared to the stuff outside of the cell is there is a bacterial chromosome, or 2. In some instances, 3. But that's where the genetic instructions are stored. And then you'll still see these little dots. Those are the ribosomes. Ribosomes are in the business of making proteins. They take the instructions from DNA molecules. Essentially, we'll talk more about this later in the class, but they take instructions that arise at the level of DNA, and they use those to string together amino acids to make proteins. The DNA, the ribosomes, the proteins made by the ribosomes all contribute to what makes the inside of a cell special. That goo, the cytoplasm, is different than what you find outside. That's pretty much the whole story. All right? Then I took you by the hand and we talked a little bit about what's up with eukaryotic cells. We got pretty far through the anatomy of a eukaryotic cell. First word that pops into my mind when I see a picture like that is, oh my gosh. All right, and now a word, complicated. There's just lots of stuff going on. That complexity that's associated with eukaryotic cells is a big stinking deal. It lets eukaryotic cells do things that prokaryotic cells could never begin to accomplish. Different prokaryotic cells might be able to do some of the things that eukaryotic cells can do. In fact, they may be able to specialize in certain things in ways that let them outcompete eukaryotes for some very specific task. But eukaryotes are polymaths. They can do everything really well in a way that prokaryotes could never hold a candle to. That complexity lets us do remarkable things. It lets us make computers, it lets us make rockets, it lets us metabolize different kinds of foods, it lets us deal with different kinds of stressors, pollutants and such that bacteria would never be able to handle. We've got a lot of stuff going on, and we use that stuff to be able to do different things. Once again, I think it distills down to this very simple idea. Structure translates to function. Structure equals function. That complexity, all that structural stuff going on in a eukaryotic cell lets us do things that we wouldn't be able to do without these structures. Prokaryotes don't have these structures. We do. We can do stuff they can't. It's as simple as that. The structures that are making that such a complicated picture are effectively internal membrane-bound compartments. We could leave the internal off. In some sense, it's kind of redundant, but these are membrane-bound compartments. They're internal because they're inside of the cell as opposed to outside of the cell. Right? So these membrane-bound compartments let us have compartments, for goodness sakes, and those compartments, the structure that's associated with those compartments, let us do lots of different things. So when we're talking about these compartments, it's helpful to stop and think for a moment about what it is a structure like a nucleus lets you do, what functionality does it give an organism that you wouldn't have had if that structure wasn't there. All right, so let's just stroll through what it is I had talked with you about on Tuesday, and we'll talk about what the functions that are associated with these different structures are. We started talking about the nucleus. In fact, I think I started talking with you about the nucleolus, the little dark spot in the center of the nucleus. I glossed over. Just a moment. Yep. So we'll do this the old-fashioned way. That's going to be bad on the Tegrity recording, I bet. All right, so works pretty well? No. Hello? Well, I tell you what, I think that's about the best I'll be able to do short of shouting, and then again, that's going to really mess up the Tegrity stuff. Well, I felt like I was on a roll this morning, guys, and now I'm literally tethered to the wall, so that's probably going to tone me down a little bit, and I'm not going to be able to gesticulate as much either. I've got this thing in my hand, but that's okay. I'm a professional. I can work with this. So I started talking with you about the nucleolus, and I kind of glossed over it on Tuesday. Let me just say a couple words about that particular structure. The nucleolus is where it is that ribosomes get assembled. So that's where the ribosomes actually get built. There are large and small subunits separately, and then they get shipped out of the nucleus into the cytoplasm where they do their thing. So ribosomes don't do any protein synthesis inside of the nucleus. Rather, the ribosomes get assembled inside the nucleolus, go out to the cytoplasm, do their thing of making proteins. And curiously, some of those proteins that they make are the proteins that you need to be able to make ribosomes. And so what has to happen is the small component parts of ribosomes have to work their way back into the nucleus, where they get assembled into ribosomes in the nucleolus, and then the ribosomes get shipped out to the cytoplasm, where they can make more ribosomal proteins, and so on and so forth. So that's the functionality of the nucleolus. It's where ribosomes get assembled. Okay? For whatever that's worth to you at this stage, that's what's going on. Moving a little bit further out, we've got the nucleus itself. The nucleus itself, gee, there's just one thing that dominates what's going on in the nucleus. It's where DNA is stored. Right? That's pretty much the only place inside of a eukaryotic cell that you find the genetic instructions that govern all the rest of the chemistry that's going on inside of that cell. Notice I said pretty much. In a little bit, I'll tell you about a couple other spots that have a little bit of DNA, but on the order of 99.99% of the DNA of a eukaryotic cell will be found in its nucleus. That's where the chromosomes hang out. So the nucleus is a structure that is particularly specialized at the function of storing and then accessing and utilizing the information that's stored in DNA. That's what's going on in the nucleus. If you like, you can think of the nucleus as the brains of the cell. And to a very large extent, what limits the size of a eukaryotic cell is how much of a distance the nucleus can exert its influence over. Right? There comes a certain point in time where you get too far out for the nucleus to keep good control over the chemistry that's going on, because diffusion can only get information to and from the periphery of the cell so quickly, and that's ultimately one of the primary things that limits the size of eukaryotic cells. So you got the nucleus. And then, boy, you guys probably still are marveling, are amazed at how obsessed I am with the double membrane thing going on for the nucleus, right? Remember I went on, what was up with that? I just kept talking and talking about how that's not just a phospholipid bilayer, that's two phospholipid bilayers. Two! How about that, right? Can you believe it? It gets a special name. It's not just a membrane, it's an envelope, because there's two of these phospholipid bilayers in play there that define the boundary between what's inside the nucleus and what's outside of the nucleus. To get in and out of the nucleus, molecules have to pass through things that are called nuclear pores. They're complicated structures. They are in abundance, but when I was talking before about the ribosomal subunits that need to move out, they pass through nuclear pores. The ribosomal proteins that get used to make the ribosomal subunits move from the cytoplasm in. So there's an enormous amount of traffic in and out of these nuclear pores. And as I'll be talking with you about later in today's lecture, when I talk about membranes, it's remarkably selective. Right? This isn't just some random sort of passage through these pores. There's a lot of regulation. Those are gates. And the gates are actively kept by gatekeepers that are monitoring the traffic and making sure the right things go in and the right things go out. Well, immediately out from the nuclear envelope is the beginning of the reticuloendothelial system. First, the rough endoplasmic reticulum. It's a good thing I have dogs, because I'm pretty good at disentangling myself from leash-like things, right? That's coming, good life skill coming in handy here. The rough endoplasmic reticulum is the first thing you see coming out from the nucleus in terms of being part of the reticuloendothelial system. Moving further out, you get to the smooth endoplasmic reticulum, and then ultimately to the Golgi apparatus. And I'll say a little bit more about each of those in a moment. But before I do, It's pop quiz time. How about that? I just told you earlier I never give you quizzes. Well, I'm starting to get into testing mode, right? I'm starting to think, what kind of question could I ask that sort of ties this stuff together? And so let me throw this at you guys, because I think this is a pretty neat question. It's the sort of thing I hope you're ready to wrestle with on Thursday of next week. So I'm asking you here a question that I think you guys can answer. There's one part here. I like this question for a number of reasons. It's actually asking you about a couple of things simultaneously, right? You can't be freaked out by the idea of radioactivity, right? It's fairly incidental to this question. It's fairly incidental to this question, but nonetheless, it's good that you have some vague familiarity with radioactivity. You have to know something about what's going on in these different structures that we're talking about, and you also need to know what UTP is. Now, I don't think I've ever actually mentioned UTP to you guys. I have talked about I've talked about uracil. UTP is the building block that's used to make RNA molecules. So it's a nucleic acid. Unlike DNA, well, which has G, A, T, and C, RNA has G, A, U, and C. And you make the RNA molecules by stringing together things that start out as ATP, GTP, UTP, and CTP. So here's UTP. It's something that gets used to make RNA molecules. With that little piece of the puzzle added, I think you guys should be able to answer this question for me. How many of you think that you'll find right after you've fed a cell, a few minutes after you've fed a eukaryotic cell, some radioactive uracil triphosphate, UTP? How many of you think you're going to find it hanging out in the nuclear matrix? Show of hands. 2? Okay. How many of you think you're going to find it in the cytoplasm? Wow, you guys are really saving your powder here for what, this is going to be cool. The rough endoplasmic reticulum. We got some takers on rough ER. That's a pretty popular answer. And how about the nucleolus? You guys like the nucleolus? That seems to be the winning answer here. I'll tell you, this is a fun experiment for all of us. I don't know what the outcome of this experiment is, right? I've not done it before, right? But I would have put my money on the nuclear matrix. I would have put my money on just inside the nucleus somewhere, right? You guys are being more specific about the nucleolus. I worry about the nucleolus because in my mind, the nucleolus is where ribosomes get made, right? That's what's happening in the nucleolus. What's happening in the rest of the nucleus is we're tapping into those genetic instructions that are stored in DNA, making RNA copies of them so that then they can get shipped out into the cytoplasm. Okay, so I'm leaning myself toward the nuclear matrix. And apparently I'm not going to put this question on the test because I don't want to have to argue with you guys. Or if I did put this on the test, I would take the nucleolus off the list of options because I could see that as being a plausible second choice. Right? But I like this question because it gets you thinking about the functionality and about what's actually happening, the dynamic of what's going on inside of these cells, given the kind of structures that you know are there. So I like this kind of question. It's not a memorization question. I like that I don't know what the right answer is. I like that I have to think my way through the answer to come up with one that I can feel kind of comfortable with at the end of the process. That's what I want you guys to be doing to get ready for this test on Thursday. Don't memorize stuff. Think about how the stuff is actually working. Okay? So, there's a pop quiz. I think there's a couple more pop quizzes coming, so don't get freaked out later in today's lecture. So we work our way out from the nucleus. I tried to get you guys armed for the next time you were in a bar or a coffee shop. I gave you this very powerful word, the reticuloendothelial system, right? I emphasized that it's a continuum. You sort of start at the nuclear envelope and you work your way out. In some sense, it peters out when you're by the Golgi apparatus, but you start off with a place in the reticuloendothelial system where there are a lot of ribosomes embedded in the membrane. What do ribosomes do? Ribosomes translate, they make proteins. Okay, so there are places in the reticuloendothelial system where it seems as if there's going to be a lot of protein synthesis taking place. That's the rough endoplasmic reticulum. What proteins do you suppose you would make there? Well, you primarily make the proteins that themselves are going to be membrane bound, so associated with membranes, or will be delivered to compartments inside of the cell or delivered to the exterior of the cell. What's left? Well, there are proteins that just hang out in the cytoplasm. Those don't get made in the rough endoplasmic reticulum. And again, I'll tell you more about that when we get to the central dogma of molecular biology in about 2 weeks. So, you got your rough endoplasmic reticulum. It blends softly and casually into the smooth endoplasmic reticulum where there is a lesser abundance of ribosomes, right? What happens in the smooth endoplasmic reticulum? The rough protein synthesis was occurring, as well as probably some other things. In the smooth endoplasmic reticulum, there are no ribosomes, so the protein synthesis is gone. That's not happening anymore. A large part of what happens in the smooth endoplasmic reticulum is that's where membrane synthesis, that's where membrane synthesis itself is taking place. So when a cell divides, you need a whole other set of membranes. Where do you make those phospholipids? You make those phospholipids in the smooth endoplasmic reticulum. Okay? And also, the proteins that got made in the rough endoplasmic reticulum, as they're moving on through from the beginning of the reticuloendothelial system to the end of the reticuloendothelial system, those proteins get modified. They get modified primarily through the attachment of sugars. It's called glycosylation. But so sugars get tacked on to the surfaces of these proteins, and those sugars can end up being a pretty large fraction of the weight of the protein and can contribute to the protein's function. Again, you're changing the structure of the protein. That in turn might have some implications for its function. I mentioned before, peripheral proteins, proteins on the outer cell membrane, often have a lot of sugars pointing out that make them sticky and account for cell adhesion, which causes one cell to stick to another cell. Okay? So, that's what's going on in the smooth endoplasmic reticulum. By the time you're through, at the very end of the reticuloendothelial system, this is pretty much where I left off on Tuesday, the Golgi apparatus. The Golgi apparatus is where the finishing touches are made for the glycosylation. There's also a lot of sorting that happens. That's what I said, sorting. In the Golgi apparatus, proteins get segregated into different little vesicles, compartments. Some proteins are only supposed to be doing their thing inside the mitochondria, which we'll talk about in just a moment. They get into certain vesicles that get delivered to the mitochondria, packaged, sorted, packaged, delivered to the mitochondria. Others only do their thing in the chloroplasts. They get sorted, packaged, delivered in little membrane-bound vesicles. Surprisingly difficult to say, I must say. They get delivered to the chloroplasts. Some proteins need to be on the exterior surface of the cell, sorted, packaged, delivered as part of these membranes in the Golgi apparatus. Some proteins end up being secreted, sorted, packaged, delivered from the Golgi apparatus. So the glycosylation gets finished, the membrane synthesis gets finished, things get bundled up and then delivered to their appropriate, hopefully, final destination. You can see in the picture here an example The artist here is trying to draw your attention to what happens. You see how the Golgi apparatus has some proteins in it, those little blue balls, apparently? They pinch off into these little membrane-bound vesicles that in turn can fuse with the plasma membrane and spit out to the cell's exterior those proteins. It's essentially what's going on with insulin and other hormones in our bodies. Cells are making those proteins. They pass through the reticuloendothelial system, they get bundled up, and then these vesicles fuse with the cell's membrane and spill out their contents to the outside of the cell. All right, so close-up picture of Golgi apparatuses. I don't want to talk with you about transface and cisface. There's an orientation that's going on, right? There's a beginning of the Golgi apparatus, and there's an end. Things move from the beginning to the end. That's plenty good enough for this kind of discussions we're having in this class. So, look at that. We've made it all the way through the Golgi apparatus. We're in the home stretch. Let's talk about lysosomes, at least a little bit. Lysosomes are internal membrane-bound compartments, separate and apart from the reticuloendothelial system and the nucleus. And lysosomes, their primary thing that sets them apart is the inside of a lysosome is just a really nasty chemical environment. Very low pH, like pH 5, very acidic. Proteins, your run-of-the-mill protein doesn't fare too well in such an acidic environment. That's why we have stomach acid. We're using the acid, we're squirting it to the exterior of our cells, essentially. There's a tube that runs through us that's outside of us to a large extent, even though it's the exterior surface of the organism in some sense. Let's leave it at that. So you use stomach acid to help with digestion to break things down, right? You're breaking down covalent bonds. You're breaking down peptide bonds because the acidity is creating the right chemical environment to break down those kinds of bonds, peptide bonds, high on the list, but glycosidic as well. The lysosomes have a very low pH, and that gives eukaryotic cells a neat advantage. They can set aside spaces, compartments within them that have a really hostile chemical environment that they can then use to break down their internal components. Say you've got a ribosome that's run its course, right? You've used it to make a couple thousand proteins. It's getting kind of tired. It's accrued a little bit of damage over the course of the time that it's been doing its thing. What do you do with this worn-out ribosome? Do you spit it out and let it go off out into the wilderness and have some bacteria eat it? Do you just say, well, I guess I'm an old cell now. I got some old ribosomes. I'm going to have to settle for living with old ribosomes floating around? No. Eukaryotic cells recycle those tired old ribosomes. They get them into their lysosomes, and then they get broken down into their component parts, their amino acids. And the amino acids, in turn, get released back out into the cytoplasm where they can be used for more protein synthesis and maybe get made back into a new ribosome or some other protein. So how about that? You know, human beings, we think we're all so swell with all this green movement and recycling. Eukaryotic organisms with their ribosomes have been doing recycling, reuse, recycle at the very least of the whole thing. I don't know about the reduced part, but they got the reuse and the recycle down for about 1 billion years with what lysosomes have been doing. So that's the job of a lysosome. Prokaryotes have a much harder time doing this kind of recycling because a pH of 5 works really well for breaking down proteins, but you don't want it breaking down just any old protein. You want it only breaking down the ones whose time has come. And so you can't have the whole inside of a cell pH 5, but if you can pull off a little compartment and if you can get the right things delivered to that compartment, you're on to something. Eukaryotes are on to this. Prokaryotes can't quite do it as efficiently and as well as we can. So I'm talking with you right now. What I've been saying is about recycling internal components, but the picture is showing you a nice example of how it is that you can use lysosomes to help break down chunks of food that have gotten into a cell as well. So it's for recycling. It's also for breaking down components of the big molecules that cells might eat. All right, peroxisomes. Not on the outline, but pretty much the same idea. A little different from a lysosome. Lysosomes are remarkable for their pH. Peroxisomes are remarkable for their abundance of oxygen-free radicals. Okay, loose cannons from a chemical perspective, making lots of strange chemical bonds, breaking things down. The concept is pretty much the same. We've got these internal membrane-bound compartments that have very special chemistries going on inside of them that would be incompatible with most of the rest of the functions that a eukaryotic cell would need to do. So we get some neat advantages from them. All right, we're really in the home stretch here. Let's talk about chloroplasts and mitochondria. So let's do it the other way around. Let's talk about mitochondria and chloroplasts. Remember the rant that I had on Tuesday? That's the word, right? I ranted I railed against the artistic license that illustrators have used for decades to illustrate the inside of eukaryotic cells. The picture at the beginning of today's lecture of a eukaryotic cell had what? 3, maybe 4 mitochondria in it, and I told you guys that's a lie. It's a bald-faced lie. Mitochondria are typically present in hundreds to thousands of copies per cell. Okay? But this is a nice picture of what one of those hundreds to thousands of mitochondria might look like. The special function that happens within mitochondria is this. Mitochondria are rock stars when it comes to breaking down sugars and extracting energy from them. Mitochondria are all about converting sugars like glucose into more useful chunks of energy like ATP, adenosine triphosphate. Believe me, you guys are going to be sick of my talking about ATP when I get to talking about thermodynamics. That's what all of the week after next week is going to be about. Okay, so ATP is just a more useful chunk of energy for a cell than the energy that's stored in a big molecule like glucose. Mitochondria, job number 1, perhaps job only, is converting sugars into more useful forms of energy like ATP. Glucose, very nice molecule for delivering a big chunk of energy from one cell to another or from one part of a cell to another part of a cell, but it's more or less the $100 bill of the energy currency that's used by cells. If you want to buy something from a vending machine, again, you guys learn a lot of strange things about me during the course of this class. My wife frequently will say to me, for some reason, I'm the only one who ever goes to the bank in my family. All right? I do all the banking stuff. But she'll frequently say to me, it's like, hey, I need some real money. Right? And what she's alluding to is the fact that my preference when I go to the bank, I like the big bills. I like to have a nice slender wallet. I love having a $100 bill in my wallet. And so when she asks me for money, right, she wants real money because she can't do stuff with the $100 bill apparently. I think she uses the money to buy things from vending machines. And $100 bills just don't get you very far with a vending machine. What she wants is she wants 5s and 10s, more useful chunks of change. That's the ATP of the energy currency of cells. An ATP is the equivalent of a $5 or a $10 bill. Glucose is more of the $100 bill. If you want to get stuff done, having a $100 bill is pretty cool, makes you feel pretty good about life, right? But having a $5 bill will probably get you further with a vending machine than a $100 bill will, right? So mitochondria, that's their job. Their job is to be the money changers for the energy currency of cell. Give them glucose, and they'll convert that very efficiently very effectively with very little overhead and banker's fees to more useful energy currency for the cell. That's what they do. Okay? Now, in terms, that's their function. Let me just point out a couple things to you about their structure. Check this out. There is an inner membrane and an outer membrane. Is there a deja vu kind of thing happening here? We've already talked about something else that had the double membrane structure. In the nucleus, it was such a big deal it got a special name. It's not a nuclear membrane, it's a nuclear envelope. Well, here for mitochondria, we got that same sort of funky thing going on. 2 phospholipid bilayers, not one, that defines the boundaries of this compartment. And there's another thing that's kind of strange about mitochondria compared to the Golgi apparatus and the lysosomes and the peroxisomes and the rough and the smooth ER. What's up with this? There's a little squiggle there with a little name tag on it. There's DNA inside of the mitochondria. There's a mitochondrial chromosome. Human mitochondrial chromosomes are, my recollection is about 200,000 nucleotides long. Puny compared to the whole nuclear genome, which for humans is at about 3 billion nucleotides. So there's not a lot of instructions there, but we've got a double membrane-bound compartment with a chromosome inside of it that contains some important genetic instructions for what it is that happens within mitochondria. Well, let's quickly go to the other compartment, the chloroplasts, where once again there's this double membrane-bound thing that's going on. Notice how they're drawing your attention to the inner and the outer membranes, inner membrane, outer membrane, inner membrane, outer membrane. And actually, for chloroplasts, inside of that double layer of membrane, there are more membranes still. Additional internal membrane-bound compartments. So the chloroplast is kind of separate from everything else. Not only does it have a double phospholipid bilayer, but inside, this is the only membrane-bound compartment that has membrane-bound compartments within it. Bags of goo inside of a bag of goo. This is new. We haven't encountered this before. The functionality that's associated with chloroplasts is the reciprocal of what's going on inside of the mitochondria to a large extent. Mitochondria are specialists at making sugars. Did I just say the wrong thing? Chloroplasts are specialists at making sugars. Mitochondria are specialists at breaking down sugars. I'm not at all sure what I just said. I had like some sort of, weird existential out-of-body experience just there. Mitochondria break sugars down. Chloroplasts build sugars up from their component parts, from carbon dioxide and water. That's what photosynthesis is all about, right? The first step in photosynthesis is you capture energy from photons. You use that energy to make ATP, the $5 bill of the energy currency, and then chloroplasts use those ATPs to make the $100 bills, the sugars, the glucose, that ultimately, when you need to get that energy back, you transport over to a mitochondria, and the mitochondria breaks it down for you. Bottom line, chloroplasts are all about harvesting and storing energy. Mitochondria are all about extracting the energy from those molecules that chloroplasts have stored them in. One other small thing. Notice that little squiggle. Doesn't show up at all in the electron microscope image, but they've labeled it for you here. Another internal membrane-bound compartment that has DNA within it. I told you on Tuesday I was going to tell you about 3 of these kind of compartments where double membrane bound, also having a chromosome. I'm probably spending more time on this with this class than you need to have me spend time with you on, but I think this is really cool. Because the single best explanation for how it is that we have these three internal membrane-bound compartments also gives us the single best explanation that we've got right now for where it is the first eukaryotes came from in the first place. There's this idea called the endosymbiont theory that plays out like this. Sometime long ago, there was a prokaryotic organism just hanging out, doing what it was supposed to do, eating stuff. One day, it ate another prokaryotic organism. Dog eat dog, bacteria eat bacteria world, then and now. Right? So this prokaryotic organism eats this other prokaryotic organism. This free-living, happy prokaryotic organism with its phospholipid bilayer, with its DNA, with all of its proteins, all the stuff that it needs to be a happy thing, but it gets eaten. And for whatever reason, the eating prokaryotic organism didn't quite finish the job. It didn't stick its lysosomes on it or its peroxisomes on it. And what we found was a prokaryotic organism, a big one, that had living inside of it a small one that was still engulfed in the eating prokaryote's phospholipid bilayer. It engulfed it, wrapped it up with its own phospholipid bilayer, and wrapped up the phospholipid bilayer of the thing that was being eaten in this double membrane. And things ended up working out pretty well, because apparently what happened is that the thing that got eaten was better at doing something than the thing that did the eating was able to do. And they were able to actually have a symbiotic relationship. One of these bacteria might have been better at fending off other bacteria or finding food or breaking food down. The other one was good perhaps at photosynthesis. Wouldn't that be cool if you couldn't photosynthesize and then you ate something and the thing that you ate while it was still alive was letting you photosynthesize? That's pretty cool. That's a neat trick for you to be able to do. Can you imagine pulling that off today as a human being, as a big complicated organism being photosynthetic? That'd be huge. So the eating organism got an advantage. Excuse me. The thing that got eaten also got an advantage. And we were on our way now to having a eukaryotic organism, where you could have specialized functions associated with these different kinds of compartments. Some later point in time, that new sort of kind of organism with the endosymbiont living inside of it ate something else that gave it another special ability. And the theory is that this proto-eukaryote probably ate three different things at some point. At one point, it ate mitochondria, something that grew up to become or evolved into the mitochondria we know today. At one point, it ate something that evolved into the chloroplasts that we know today. What's the third thing? It ate a nucleus. We have these internal membrane-bound compartments that each have these specialized functions. One of those specialized functions is the storage, the maintenance, the accessing, and the use of genetic information. One of those internally eaten endosymbionts, one of those symbiotic organisms that got eaten, became the specialist that we know today as the nucleus. And it got to be so good at storing and maintaining genetic information that the chloroplasts' genes and the mitochondria's genes all turned out to be better stored in the nucleus. And even the organism that did the original eating said, gee, nucleus, you're better at this than I am. It shipped its DNA over into the nucleus for the most part. So that's what's known as the endosymbiont theory. The best proof that we have for it is simply this fact that we've got these three double membrane-bound compartments, each of which has some chromosome material associated with it, and the fact that we can see this happening to this day in some blue-green algae, for instance, where they're picking up things that effectively give them the ability to be photosynthetic where they weren't before. So anyway, that's the endosymbiont theory. It's time for a test on the endosymbiont theory. So according to that theory, which of the following organelles was once a free-living organism of its own? A, mitochondria and lysosomes. Lysosomes are a loser there, right? B, mitochondria and chloroplasts. Okay, we're done, right? All the other answers are wrong. Okay? Now, I could have substituted nucleus for mitochondria or chloroplast, and I think that would still be the right answer. But nonetheless, that's a good answer right there. B is the one that I like. All right, cytoskeletons. So there are these structures inside of eukaryotic cells. I use the word structure. When I say structure, that means function's going to follow. Eukaryotic cells aren't just bags of goo. They're bags of goo with a matrix that holds the bags into certain positions relative to each other. That matrix is the cytoskeleton. And here's a picture that shows you the different kinds of things that can happen here. You notice all these bluish, green sort of stripes? Those are tubulin molecules that have been lit up, dyed with a particular kind of molecule. These orange ones are actin, a different kind of tubule that cells use to maintain structures, lit up with a different kind of dye. And still another kind of protein is lighting up the nucleus of this cell. Check it out, right? When you see them all lit up together like that, you can see that, yeah, well, there may be mitochondria, and there may be chloroplasts and reticuloendothelial systems, but you got this network of a framework on which they're all hanging. So let's look at them one at a time. Just very quickly, here we're using that green dye to light up for you a particular kind of cytoskeleton component called tubulin. And you can see these particular eukaryotic cells, the whole cell lights up with this network. It's a three-dimensional grid work of tubulin that gets assembled in this way to give you this grid work that you can hang all the other components of the eukaryotic cell on. Here's another dye that's lighting up for us here, actin. Actin tends to be more peripheral than the tubulin is, but it's got a different structure. It's made from different proteins, and it helps give the three-dimensional exterior shape of the cell. My point here is simply this, is we've got these networks of proteins, these fibers of proteins that give all of our eukaryotic cells structures. And then there are even these things called intermediate filaments that are a little bit bulkier and more rigid than the tubulin and actin fibers are. You also got these other structures in eukaryotic cells. All eukaryotic cells have centrioles, remarkable structures in their own right. When you see the electron microscope, you can see the picture here is actually doing a pretty fair job. They're always in pairs, and they're always at a right angle to each other. Later on in the class, I'm going to be talking with you about genetics, about how it is that Mendel's laws all work out, cell division, those sorts of things. Centrioles are going to come up again then. But there are some pictures of these structures. Every cell will have at least two. Sometimes they'll have four, depending on what stage of the cell cycle they're in. They're always at a right angle to each other. They have this interesting arrangement of microtubules that you find only in centrioles. And something that doesn't often get said about centrioles is that they contain some DNA as well. Best guess from an endosymbiont theory perspective is that's what's left of the original prokaryote's genome, the one that did all the eating in the first place. All the rest of its genes got shipped over to the nucleus, but there's still a couple vestiges left that are associated with the centrioles that end up playing an important role with cell division. Lastly, isn't that a cute picture of a cell? I don't know, it's kind of fuzzy. It looks like you want to pet it. It's, There are these things that poke out of cells as well. This one's fuzzy because it's got all of these things poking out. The things poking out here are cilia. They help with motility. Boy, when you start to crack open the inside of a cilia and look at what's going on there, it's a wonder to behold. These are pretty elaborate structures, and yet every organism, prokaryote and eukaryote, that uses cilia has exactly this same structure. So when you're looking at it, I would be thinking, okay, so that one's pretty. Any cilia that you sliced longitudinally, you're going to find this arrangement of triplets that are held together on this central spoke. All I really want you guys to walk away with from looking at this picture is this, that there's tubulin involved, right? So these are structures that are associated with the cytoskeleton, and they're pretty elaborate, pretty complicated structures. The fact that that complexity is uniform across all of life, archaebacteria, prokaryote, and eukaryote, suggests that cilia got worked out very early in the history of life. It's a structure that works. It works well. Don't mess with it. For over 1 billion years, living things haven't messed with this structure. And also, at the periphery of the cell, we've got all of this collagen and fibronectin and proteoglycans, the glycocalyx taken as a whole, that gives you this pretty impressive network of stuff that's going on. And I'm going to say more about that now as we transition from this lecture outline to the one that I was going to talk with you about on Tuesday. That's next week. So, let's focus our attention for the next 15 minutes or so, maybe 20, yeah, give me 20, on plasma membranes. Right? I'm not going to bother you anymore with nuclear envelopes or that sort of thing. Let's just talk about plasma membranes. And if you want, we can really focus this just on the exterior membrane of the cell, the cell's outermost membrane. And let's talk about its structure, and then from the structure, figure some things out about how it works, what its functionality is, what it can and what it can't do. So here again is that picture. This is a modern conception, at least for this artist, of what it is a plasma membrane looks like. Right? The star of the show is the phospholipid bilayer. Phospho here, these little heads, lipid, these yellowish tails, bilayer because you got a phospholipid here, you got a phospholipid here, one layer, two layer, mono, bilayer. All right? Associated with this phospholipid bilayer are a bunch of proteins. On the outside of the phospholipid bilayer is a watery environment where there are a lot of proteins, and a lot of these proteins have sugars, these green things, attached to them. On the inside of the phospholipid bilayer, inside the cell, it's a watery environment, and there are a bunch of proteins present that give some structure, some rigidity to this three-dimensional shape of the cell. Those microfilaments, the tubulin, the actin that we had just been talking about. That's what we think of when we talk about plasma membranes today. Right? That's the state-of-the-art understanding of plasma membranes. It wasn't always that way. And I mentioned Davison and Danieli, excuse me, largely because I think it's illustrative of how our best understanding sometimes changes. Right? There's a lesson to be learned from the story of Davison and Danieli. When I took a freshman biology class many decades ago, Davison and Danieli was the model of the phospholipid bilayer. This is what I was taught plasma membranes look like. Turns out Davison and Danieli were wrong. And they were wrong for about 50 years, right? In the 20s, they came up with this Davison and Danieli model. They got tenure. They sat fat and pretty and collected a salary and didn't do much teaching, but got a lot of grant support for about 30, 40 years because they were the folks who came up with the Davison and Danieli model for plasma membranes. Now, they were wrong, but here's the way that they were thinking. Their thinking wasn't way off left field. It was just wrong. And you guys now will know why it is they're wrong, whereas it took 50-some years to correct them. But here's their thinking. They knew from folks that had been doing work in the late 1800s that cell membranes for a eukaryotic cell, and specifically for red blood cells, they knew that things that were hydrophobic had an easier time getting from the outside of a cell to the inside of a cell than things that were hydrophilic. Right? So they had that little bit of information from the late 1800s, and they were thinking to themselves, gee, what are cells made of? There's a lot of these phospholipids, there's a lot of these proteins, there's some nucleic acids, there's some sugars. What's the boundary of a cell? Using that one little clue, they got the idea that plasma membranes were likely to be phospholipids. That explained why hydrophobic things could get through a cell membrane more easily than a hydrophilic thing, because most of the membrane was phospholipid, right? And so that made it permeable to lipids more than it did to water-soluble things. And they knew something about the structure of the phospholipids. They knew that the phosphate was about 10 atoms at the top. They knew that the phosphate was about 10 atoms at the bottom. And they knew that the lipids were about 20 atoms long each. Put it all together, and you get something that's 50, maybe 60 atoms thick. And so the Davison and Danieli model took these phospholipids, arranged them as a bilayer, and they also knew from cracking open cells and isolating just the membranes that the membranes also had proteins associated with them. So where are the proteins going to go? Well, here's where they went astray. They didn't appreciate that proteins could be hydrophobic themselves. All the proteins they had ever encountered in their undergraduate laboratory experiences were water soluble, right? So they didn't appreciate that proteins could be fat soluble, could be hydrophobic. They knew membranes had to have some proteins associated with them, so they had their 50, 60 atom thick phospholipid bilayer, and they put the proteins on the top and the bottom, and they made a really nice Davison-Danieli sandwich. Protein on the top and the bottom is the bread, phospholipid is the meat in between. That's the Davison and Danieli model. What ultimately demonstrated that they were wrong is when folks were finally able to start using electron microscopes to measure the thickness of actual membranes. Remember, the Davison and Danieli model is saying 50 to 60 atoms from here to here, and then the proteins, we don't know how big they're going to be, but that's going to be at least another 20, 30 atoms here, another 20, 30 atoms here. Membranes, they're predicting, would be over 100 atoms thick. Electron microscopy revealed that membranes were actually about 50 or 60 atoms thick, just the phospholipid thick, without the bread, only the meat, none of the bread for this sandwich. And that, in turn, caused a pair of other guys, whose names were Singer and Nicholson, in the 70s, to put forward a different model to the Davison-Danieli model. They called their model not the Singer and Nicholson model because they didn't want to have people poking fun at them someday the way that they poked fun at Davison and Danieli. Rather than putting their own names on this model, they called their model the fluid mosaic model. And the picture of what their model looks like says pretty much what it is the fluid mosaic model is. The principal difference between the fluid mosaic model and the earlier Davison and Danieli model is that now, instead of having the proteins only on the top and the bottom of the phospholipid bilayer, here the proteins are embedded within the phospholipid bilayer. that's pretty much the basis of what we think plasma membranes look like these days. So that's the structure. Okay? Let's talk a little bit about, zoom in on the structure, and then we'll talk a little bit about the function. So you got these proteins, right? These big bluish, purplish blobs here are proteins. 1 protein, 2 protein, 3 proteins. There's another one bobbing around over here. You guys know that if you used hydrophobic R groups to make your protein, this part of the protein, if it has a hydrophobic R group, is going to be pretty comfortable hanging out in this hydrophobic part of the phospholipid bilayer. But you have a big enough palette, a rich enough palette when you're making proteins, that you can have some protein parts that are made from amino acids that are hydrophilic. That can poke out at the outer surface. You could have others that poke out at the inner surface. And here is another zoomed in sort of view of what one of those phospholipid bilayer proteins might look like. This is a membrane-bound protein. You guys recognize this coily thing here? It looks like a ribbon. That's an alpha helix. 1 alpha helix, 2, 3, 4, 5, 6, maybe 7. Right? If this protein got made with a primary sequence that caused it to fold up into those types of alpha helices, and those alpha helices further had hydrophobic R groups associated with them, those alpha helices would be happy interacting with each other because they're excluding water, and they'd also be happy interacting with the lipidy part of the phospholipid bilayer. This corresponds, it's an artist's rendering, but that corresponds to what an X-ray crystal structure of that protein would look like. That's how many proteins that are membrane-bound actually look. They use those alpha helices to give them something that effectively anchors them in the lipidy part of the phospholipid bilayer. And then other parts, here the beginning, here the end of the protein, poke out. This part pokes out to the outside, this part pokes out to the inside. These parts are going to be made with, there's a beta sheet, right? These parts are going to be made with amino acids that have R groups that are hydrophilic, water-friendly. Okay? So, I just told you the answer to this one here. Third and last quiz for the day. Which of the following amino acids would be most likely to be present in the transmembrane domain of an integral membrane protein? So there's some jargon there. Let me backtrack. This is an integral membrane protein. Integral in the sense that it is integral to the membrane. It's part of the membrane. And the transmembrane domain. This is a transmembrane domain. That's a transmembrane domain. It's transmembrane because it spans the membrane. It's a domain because it's essentially an alpha helix. So what kind of amino acids, I'll translate the question. What kind of amino acids do you expect to be part of the protein that's nestled up against the lipids in the lipidy part of the phospholipid bilayer? Right? And I like this question too, because it's pinging you on a couple of different things. Right? You have to know a little bit about the jargon, but you also have to know something about hydrophobicity and hydrophilicity, and maybe a little bit about amino acids. So I'm not asking you to know that lysine is a charged amino acid. That's gratuitous. That doesn't need to be there. But seriously, anybody think a charged amino acid is going to be hanging out in the transmembrane domain? If you think so, we need to talk, right? Because that's just not, that doesn't work, right? How about a polar amino acid? Well, that's a little more likely than a charged amino acid, but still not going to work. A special amino acid, like proline. Proline is special in the sense that it introduces bends into proteins. That's what makes it special. It's got nothing to do with hydrophobicity or hydrophilicity. Any of the above? Well, I hope not, because these three are pretty bad answers. we're pretty much just left with this choice over here. How about a hydrophobic amino acid like valine or leucine or isoleucine? Any of a number of the hydrophobic amino acids, that's the kind of R group, that's the kind of amino acid you would expect in the transmembrane domain. All right, on Tuesday, I waved my hand at this idea a little bit. I can talk about it in a little more detail now. Making little knots here. The kind of movement that happens, and we're getting into function now, the kind of movement that happens in a membrane is governed heavily by the structure of the membrane. It's really as simple as this. Realize that that's intensely negatively charged, that phosphate group, and that's intensely negatively charged. It is really hard to drag this negative charge away from all of its watery friends up here through all of this very hostile territory of hydrophobic chains of fatty acids, or fats really, lipids, all the way through until it gets out to the other side where it can make some new watery friends. That flip-flopping is just darn difficult. So difficult that for a typical cell, it happens on the order of once a month, right? It's just not worth talking about. It just doesn't happen with any appreciable frequency, especially in comparison to the sort of lateral movement, swapping spots, one phospholipid swapping its place with another phospholipid in the plasma membrane. That sort of side-to-side, front-to-back kind of movement, there's nothing that stops that from happening. In fact, it happens for any given phospholipid about 10 million times every second. That's lots of jittering around for these molecules. So again, the structure here is going to impart something important to the functionality. These phospholipid bilayers are really very stable in the sense that they're bilayers, but not very stable in the sense that there's a rigid layer with one phospholipid linked to another. And so just as it's hard to move these guys flip-flop wise, it's hard to drag a water molecule from over here through to over here because it's got to pass through this hostile territory. The way that cells get things from outside of the cell to inside of the cell, and vice versa, is almost always mediated by specific proteins. So the last little bit that I want to talk with you about today is what's going on with these kinds of proteins. And we find there are a bunch of different kinds of proteins. They're giving you essentially 6 different categories of proteins that are embedded in these phospholipid bilayers in this fluid mosaic model. There are those that are involved with transport. There are those where the enzymes are just hanging out in the phospholipid bilayer but involved with some sort of catalysis. There are those that span the membrane and have one part that's dangling out on the outside waiting to receive a signal, and when the signal is received, ends up changing its conformation so that that information can get passed to the inside of the cell. There are proteins that are involved in holding cells together, or in letting one cell type recognize another cell type. And here again, another kind of intracellular holding together. And there's things that cause cells to get anchored to other things like bones, for instance. All right? So, in light of what you guys now know about the fluid mosaic model, here's an experiment that goes back about 100 years that I think you guys can predict what the answer is going to be. So the question here that we're testing is, do proteins move when they're embedded in a membrane? And the experiment is going to be this. We're going to take some mouse cells and take some human cells, which have, I hope you appreciate, different proteins on their surfaces. Here, conveniently, we're showing the mouse cells as red and the human proteins as blue. mouse proteins, human proteins, and we can fuse them together. We can smush them together and then ask, what will we see? If the proteins do not have lateral motion, the way that the phospholipids do, then the fused cell, the hybrid cell, should have one part that's all blue proteins and another part that's all red proteins. But instead, what you see is after a little bit of time, those proteins just bob around in the phospholipid bilayer and they diffuse and they spread around throughout the entire surface of this hybrid cell. Again, now, this experiment again illustrates that you can have that kind of lateral motion of proteins. Okay, we're definitely close to the end. We're pretty much down here talking about permeability. Well, here's the exterior of the cell. Again, just ooh, right? There's a lot of stuff going on outside. It's not just a matter of proteins spanning, but there's all these other glycolipids and such that are causing them to be sticky. Let's talk just a little bit. This is hearkening back to some things you did in lab last week. Let's talk just a little bit about semi-permeability. Permeability means that things can move through something. We're talking about membranes. So a permeable membrane is a membrane that stuff can move through in either direction. Anything can move through in either direction. Not much of a membrane, not much of a boundary. But semi-permeable, what that means is that some things can move through, maybe in just one direction and not the other, and other things can't move through. Our plasma membranes, the plasma membranes of cells, are semi-permeable membranes. So here's what happens when you're talking about just a plain permeable membrane. This membrane is letting water go this way, and it's letting water go this way. And eventually, what will happen is you'll get the same number of water molecules on both sides of the membrane. If you find yourself in a circumstance where there are more water molecules on one side than the other, what that translates to is there's more opportunities for molecules to work up a head of steam and move from this side to the other. Right? So again, eventually you work up to a situation where you have equilibrium, the same number of molecules on both sides. Check out what happens when you've got a solute in here. If you have water on one side and say a sugar on the other side, what will happen is that both of those molecules will reach an equilibrium where you should get the same number of water molecules and sugar molecules on both sides. Here's another pop quiz. I thought I was done with quizzing you. Let me ask you this one quickly. Which of these molecules will get across a phospholipid bilayer the easiest? Water, bad choice. Oxygen gas, that looks like a winner to me, right? Because this should be fairly fat soluble. There's no partial separation of charge. Phosphate group, bad choice. Sodium, bad choice. Glucose, bad choice. Lots of charge, lots of polarity there. But check this out. I want to share with you a personal story. I think I got enough time to pull this off. When I was a freshman in college, I had 3 nights running where I didn't sleep because I was so excited about a discovery that I had made. I thought I had figured out how to do perpetual motion. All right? What I wanted to do was this. I tell you what, I figured, let's see if you guys can come up with a perpetual motion machine with the idea in this slide, and I can tell you about it on Tuesday during that review session. I won't take time to do that here. I do want to tell you about active versus passive transport. It's simple, right? Proteins are the gatekeepers of the membranes. It's the proteins that are letting molecules go from one side of the membrane to another. Different proteins can be engineered. They can be designed in a way that it makes it comfortable for glucose to move through, but not fructose and not an amino acid, right? So passive diffusion is when, or passive transport is what's happening when you're simply going down a concentration gradient. Active transport is when the cell is using these proteins, but pulling molecules from low concentration to high concentration. To do that, they have to expend energy. They typically do that by burning ATP. So there's passive transport and active transport. Both are mediated by proteins. Both are what's helping us get stuff from outside of the cell to inside of the cell, and sometimes from inside of the cell to outside of the cell, depending on the cell's need. All right, now you can go. See you guys on Tuesday. Remember, a week from today, we'll have our first test.